mummyberry and botrytis, and I'd love to welcome our first speaker, Dr. Dalphy Harteveld. She is a postdoctoral research associate in WSU Mount Vernon, and she's going to speak about mummyberry on blueberry in the Pacific Northwest. Please welcome Dr. Dalphy.
Hello, thank you for this introduction, and thank you for attending my talk today. I've been in Mount Vernon at the Washington State University Northwestern Research and Extension Center for about 2 years now, and my research has been focused on mummyberry. So today I'm going to present what I found during those 2 years. And so I'm a postdoc with the Department of Plant Pathology, and I'm supervised by Dr. Tobin Peaver. The research was funded by the Washington Blueberry Commission and the Washington Commission on Pesticide Registration. So I'm first going to give you some overview of what the disease mummyberry looks like in the field and how you can find it in the field at different times in the year. So here you see photos of the typical flower strikes, which are the symptoms you can observe in spring. And you see the brown discoloration of the floral tissue with some fluffy mycelial growth of the fungus on the surface of these samples. Here you see some more photos of these flower strikes later in the season when the fruit already started developing, and you can see the remainder of the infected flower clusters still on the plants. This is the second strike symptom you can find. So the leaves get infected where first there's some brown discoloration of the leaf veins, as you can see in the top left photo. And then the browning expands over the surface of the leaf blade, and this fluffy growth on the surface of the sample starts to show, eventually taking over the whole leaf cluster. Now this disease or this fungus has a very cool aspect in that it can reflect fungal growth reflects UV light in such a way that it attracts insects, including bees, and these land on these symptoms, and the spores called conidia are attached to their furs and legs, and they take them with them into the flowers, the open flowers, where these spores are infecting the flowers and colonize the styler, canal, and ovaries, infecting the fruit from within. So here you can see some photos of fruits of different stages. So from green fruit at different stages of ripening, and you can see that when you cut them open, you can't always see from the outside that the fungus is apparent inside. So here on the bottom photo, on this green fruit, you see when it's cut open on the right side that you can see the white star-like figure in there representing the fungal growth that is accumulating within the fruit. So if early in the season you want to know if you have mummy infections occurring, you can cut open your fruit and see if you have mummyberry that way. Then later in the later over the ripening stages, the fruit turns pink and starts to show this like segmented discoloration with pink and blue, and that's the mummifying of the fruit taking place. And here you see some more photos of the fruit symptom, how it appears on the crop, and you see the light pinkish fruit that doesn't turn into blue or purple fruit, but just stays pink and eventually turns white, shrivels, starts forming a pumpkin-like figure or structure, like in these photos, and eventually drops off the plant. Now, once it's on the ground underneath the blueberry plants, the so-called mummies turn black and form more of a swollen, like pumpkin-like shape, as you can see in these photos. And you can find them when you remove the leaf litter underneath the blueberry plants. where they reside, staying about halfway through the, halfway sunk in the soil. Then early spring, when the conditions are right, there's these mushrooms called Apothecia developing from these mummies. These look like this. They first, here on the bottom left photo, you can see how small they are. So when they start forming these Apothecia, first with very small openings at their terminal end, and these opening up wider and starting to produce spores, which are called Ascospores. Now, this Ascospore release is the release of these initial spores, and it's like even up to millions of spores per mushroom. And to give you an idea of what that looks like, I obtained this video from Philip Wharton at Michigan State University, and I'm hoping that it's gonna work. Let me see. So yeah, here you see the typical Ascospore release of the Apothecia. So here, like under the, once there's a wind flux or changing conditions, you have this sort of puff of spores being released. So I showed it one more time. So there you go. Now, and okay, so mummyberry has been a problem in the US Pacific Northwest, causing fruit yield losses of up to 70% in individually affected fields. The curing control methods are inconsistently in controlling this disease, where the timing of fungicide applications is based on a routine calendar-based schedule rather than based on the biology of the fungus. And there's limited information on the biology of this fungus in this area. And most research has been performed in Eastern North America, because this disease is occurring in all areas where blueberries are grown. But most research has been performed in the other states and not in the mild climate that we have in the Pacific Northwest, which may be the reason that we can't use the methods that they use to control this disease in other areas. So here you see again the disease cycle that I just worked through with each individual steps. And to protect your crop from this ascospores to infect the tissues early spring, the control method should coincide with this ascospore release. And so the crop can be protected from these ascospores early in the season, and therefore prevent the disease to further accumulate. So this was the stage that I was focusing on during my research, the Apothecia development and Ascospore release. So looking into that a little bit more, here you see a graph that shows the timing that this Ascospore was, or the Apothecias were found in different areas. And you can see that in different areas, the reports show different timing that these Apothecia were around, from beginning of March to end of March. And then looking at data obtained from Corvallis in Oregon, from Professor Jabe Scheit at Oregon State University. Here you can see almost 10 years of data of this timing of these mushrooms being around. And you can see that the timing was very different from end of March till mid-April, which makes it hard to plan the application of fungicides accordingly. So looking at the process that happens from the fruit infection to the next stage of Apothecia production. So these mummies go through several stages, from maturation to dormancy, germination, and then the actual Apothecia production and sporulation. And what we know through research is that to break the dormancy period of this fungus over winter, it requires a chilling requirement of a period of time with temperatures between 0 and 7 Celsius. And for the next stage, from germination to the Apothecia production, it needs a certain threshold of environmental factors. Research performed mostly in the eastern side of North America showed that this chilling requirement was different in different regions, where 900 to 1200 hours of chilling were required in the high bush blueberries grown in North Carolina, but it was less than 700 hours in the rabbit-eye blueberry in Georgia. And in the southern Washington, it was about 700 hours in high bush blueberry found by Pete Bristow a couple of years ago, and indicating that the climate in the Pacific Northwest may indeed provide different effects in this disease cycle of this fungus. Now, environmental factors that have been indicated previously by different researchers were relative humidity, air temperature, soil moisture, and light that have an effect on this Apothecia development. However, these studies didn't implement all the factors together to see which ones are the most important in affecting this Apothecial production. And there's a lack of studies that assesses this in an in-field situation rather than laboratory experiments just focusing on a single factor. Now, then another aspect of infection risk of this Ascospore release occurring is that it has to coincide with the timing that the host is susceptible. And so the blueberry plants are only susceptible when young emerging tissue from flower and leaf buds is available. So only at the early springtime that these tissues are available. And previously, the recommendations for the timing of fungicide applications was to time your fungicide application based on the leaf bud development when they start breaking open and show the first images of the green tissue appearing to coincide your fungicide sprays with that to protect the crop. Now, what previous research also indicated is that there might be host resistance through the phenology of the plants, where you have early maturing cultivars and late maturing cultivars that therefore might have a different time of when they have this first bud break occurring. And if that bud break is missing the time that the ascospores are produced, may therefore be providing some source of resistance. Now, based on this information of other researchers, I developed 4 objectives for my research, where the first one was to determine the timing of primary inoculum, so this ascospore release in the Pacific Northwest, to determine the environmental factors that influence Apothecia development as well as longevity, because we want to know when it starts, but we also want to know when it ends, so we know how long we need to protect the crop from the ascospores being in the air. Now, the third objective was to determine differences in timing of bud break and therefore the susceptibility of blueberry cultivars grown in this area, and to develop a prediction model to predict the ascospore release in this region. So during this research, I approached this as the total ascospore release as this triangle of 3 different factors that need to be determined, giving a better idea of what this ascospore release does in this area. So starting that this is a combination of the pathogen, the host, and the environment. And now, so the first objective I'm going to talk about is the timing of primary inoculums by the pathogen. So I did field experiments during the 2015 and 2016 production seasons, where I monitored the Apothecia development in 3 fields, 1 field in Whatcom County, 1 field in Skagit County, and 1 field in Benton County in Oregon, where Jape Scheit was providing the data for. And I placed Burkhardt spore traps, as you see in this picture on the left side, to capture ascospores out of the air. Now, in this graph represents the results of this experiment, where you see the different colors indicate the different fields, and the different patterns are the 2 different seasons. And so the blue and the green bars are the Washington fields. You can see spore release was found from beginning of March till almost the 2nd week of April, which is very long. It was almost 40 days, which is almost on the maximum side of the ascospore release timing that's been reported to date indeed indicating that this area is very susceptible or conducive for this disease to occur. You see that in Oregon, the timing was slightly later than in Washington, and that there's a big difference between the 2015 and the 2016 season. And this may be caused by the different soil type that they have there. It's like in Oregon, they have more of a sandy type soil that dries out quicker than the soils that we have up here. And if the moisture of the soil goes down, it also may provide a less conducive environment for Ascospore release and mummies to germinate. Now, moving on to the environment and what environmental factors have an effect on Apothecia development. So I used two weather systems. I used the WSU AgWeatherNet data that is publicly accessible and has weather stations all across the state and other states. And then I placed in-field weather stations in the three fields that I've been working in, measuring air temperature, relative humidity, leaf wetness, and soil temperature. And these were indicated in red here, measured in both systems. Then in-field weather stations also included soil moisture data, and these precipitation and solar radiation data was also included in the analysis from the AgWeatherNet. Now, the station of AgWeatherNet that were included in this study were about 1/2 mile to 2 miles away from the field locations used in the study. Now, in this table, it shows the result of the analysis of each of these weather factors. And in this table, it sort of shows that the stars indicate the level of significant impact they had. And you can see that already just looking at two different time periods. One was used to look at the timing, the factors involved in the Apothecia development, whereas the second time frame that was post the Apothecia production, initial Apothecia production, was looking at how these factors influence the longevity of this Ascospore release production to occur. And you can see that pretty much almost all the factors had some significant impact. However, soil temperature, air temperature, leaf wetness, solar radiation, and soil moistures were the 5 main factors that have a significant impact on Apothecia development and longevity. Now, to get an idea of what the values or thresholds of these all these 5 factors were, here you see in the table the average calculations of these factors for a month before, during Apothecia development or presence, and a month after Apothecia being present. And you can see that about temperatures of about 50 Fahrenheit were conducive to Apothecia presence, 43% of soil moisture, 12 megajoules per square meter of solar radiation, and a 0.3 unity of leaf wetness. Now, so these might be factors that you might that you can look up on either the WSU AgWeatherNet or other means, and give you an idea of if you are close to reaching those threshold values for Apothecia production. Now, what we also found, comparing this WSU AgWeatherNet system versus in-field weather stations, is that the AgWeatherNet data can be used instead of in-field weather stations, and is detailed enough. However, I'd like to say that we never used stations further away than 2 miles from the fields that we were assessing. And soil moisture data was not included in the AgWeatherNet systems. So soil moisture may still need to be measured in the field sites that you're looking for. Now, moving on to the third factor in the infection risk system, which being the host. Here you see an overview of the different flower bud and leaf bud stages that are accumulating in the field throughout the season. And based on literature, bud burst and tight cluster stages are the susceptible stages for infection, as well as late green tip, which is the stage where over a quarter-inch of green tissue is available. And what I was curious about is if the timing that these susceptible stages is present on the crop in this area at different times or at the same times, and if the cultivars are therefore changing the window of susceptibility to this Ascospore release. So in both Whatcom and Skagit County, I selected 3 fields of 4 cultivars, which being the blue crop, Duke, Liberty, and Draper. I marked 6 shoots on 6 plants per field, and I monitored weekly, taking photographs of the shoots and determining the stages of the shoots from these photographs. So in these graphs, you can see the percentage of susceptible flower buds during Ascospore release. So the time frame that is presented on the x-axis of these graphs is the time frame that we found that Ascospores were being produced. And the y-axis is the percentage of these susceptible stages found on the plants. Then the continuous lines and dotted lines were the difference between the 2 counties, and the different colors are the different cultivars. Well, and what you can see here is that the blue and green, so being blue crop and Duke, had their peak amounts of susceptible flower tissue on the plants earlier compared to Liberty and Draper, and therefore showing a potential shift there that if Ascospores release might have occurred sooner than that, those would be the most susceptible cultivars. However, now in this scenario where Ascospores are released, the yellow lines being Liberty show that Liberty would have had the most, the highest amount of susceptible flower tissue on the plants during this period, being therefore more susceptible. Now, and these are similar graphs, just representing the leaf buds. And there was a lot more variation in the between the cultivars and the counties, showing the peak numbers of susceptible leaf buds on the crop. However, you can see that blue crop, being the blue lines in the graphs, had its peak number of susceptible leaf buds earlier in the season compared to the other cultivars, and to the extent in 2016 that blue crop was already coming down from the high numbers of susceptible tissue compared to the other cultivars, and therefore potentially avoiding infection potential from the fungus. Now looking at so these graphs represent the total amount of susceptible tissue on the plants over the whole season. So this is calculated using a formula modified and called area under susceptible tissue curve, and is a one value calculating the entire surface under one of these graphs, just to give you an idea of what this value might mean. And on the left is the total value for flower tissue, on the right for leaf tissue, and you can see that the amounts of susceptible flower tissue were a lot higher compared to leaf tissue, which is an important finding, especially seeing that previous recommendations have always been focused on vegetative buds for the timing that the fungicides should start. Whereas now we see that the flower tissue might actually play a more important role, and you can use it as an incentive to pay close attention to both bud times. So yeah, I now would like to just recap what I've just been talking about and what the outcomes of this part are so far, and what it is meaning for this area and compared to other research. So we found that we are experiencing relatively long periods of Ascospore releases in this area, which up to 37 days, occurring between early March and mid-April. We found that multiple environmental factors are associated with Apothecia development, and this includes soil moisture, soil temperature, solar radiation, air temperature, and leaf wetness, and that flowers may play a more important role than leaf buds. leaf buds. Now, what does this mean and how can we use this information is that having the long period of Ascospore release, and we know that we also need to, we can expect that we need to apply protection of the plants for a relatively long period as well. And that we can time these fungicide applications and estimate the duration that control measures should be applied based on these weather factors, which can be monitored by WeatherNet or in field weather stations that can be established, and that both flower and leaf buds needs to be monitored closely. And often what I found from this research in the field is that flowers develop earlier than leaves, also reaching their susceptible stages sooner than the leaf buds. So therefore, it's important to pay close attention to both flowers and leaves. Now, moving on to my fourth objective, which is the prediction model for Ascospore release, pulling these three pieces of research together. Well, here is the approach we took to build a prediction model. I've been doing this work in collaboration with Michael Grant at the University of Washington. And so we use this Apothecia value as a binary value of like presence and absence, and looked at the data collected for several environmental factors and included this in a database or data frame, looking at the data period from 1 month before the Ascospore release period and 1 month after, including about 60 days of data. We split all this data into a 70% training and 30% test set as a validation process, an internal validation process of all this data, where this 70% training data was used to develop a model and test algorithms on this testing data set, and with that performing an internal validation. And this was done 500 times. And to test different models using this system, I picked 4 different models to apply this model building technique on. And here you see results of this experiment, where the 4 different models are indicated at the bottom of this graph. And GLM is a generalized linear model. I won't go into the models too much, but we have one model on that GLM model that is more of a traditional method to build prediction models, whereas the 3 other models on the side are modern techniques that have been coming with the machine learning and all the computational analysis strategies that we have right now. And you can see, so in the different colored boxes is the numbers of predictors. So the numbers of environmental factors included in the analysis. And on the y-axis is the level of accuracy, where you see if it's a 0.8 level of accuracy, it means 80% of the Apothecia were predicted correctly. So now between the older method and the newer methods, you can see that the newer modern prediction models are having a higher accuracy compared to the traditional methods. And that the random forest was performing the best with a performance of about 80% accuracy in predicting this Apothecia correctly. Now, the number of predictors that could be included in this model could be reduced from 7 to 3 factors only. So, and these 3 factors were soil moisture, soil temperature, and solar radiation, indicating that we can just limit our factors that we need to pay close attention to for Apothecia prediction to only those 3, instead of looking at all the factors that I talked about before. So here I point those out a little more. So the 3 most important environmental factors were soil temperature, soil moisture, and solar radiation. The range of these in the conducive environment of Apothecia production and average are indicated here on the right, where for the temperature, it was a range between 43 and 57 Fahrenheit with an average of 50. The soil moisture between 20 and 100%, being average about 40% soil moisture, and the solar radiation between 2 and 22, with an average of 12 megajoules per square meter. And the susceptible host stages of being flower bud break and tight cluster and quarter-inch green, these are the factors that should be included in the overall prediction of Apothecia release and the infection risk of crop in your field. Now, and to put that in sort of a practical application that could be used. So here, this is an overview of how you can estimate risk in your own field. So you need to get an indication of if there is history of mummyberry in your field or in neighboring fields in your area. If you need to have a good understanding of if you have early or late maturing cultivars, and monitor the bud development of both flowers and leaves for them to reach these susceptible stages. And you can check in-field weather stations or AgWeatherNet data for the thresholds of soil temperature, soil moisture, and solar radiation. Now, I'm currently working on an app where all this data is going to be included, and I was really hoping to present that today, but I didn't finish it just yet. But I can give you a idea of what it might be looking like. So here on the left, you see the 3 weather factors that were included in this prediction. So temperature, so moisture, and solar radiation. Then you can insert the values that you've been measuring either with your sensors in your field or with AgWeatherNet data, and insert the stages of flower buds and leaf buds that you observe in your field. And these are like drop down menus where you can pick your stage, and you press update, and then a prediction will pop up. That here I just printed a prediction that is based on a heart disease, but this is somewhat what it's going to look like. It will give you an estimate of if your field's at risk for Apothecia being there or not, and what the probability is of how close it is that these are being produced. So, and yeah, once this is finished, I will be notifying everybody via either the Washington Blueberry Commission and put an article in the newsletter and update you with this information, hopefully also establishing some sort of workshop or yeah, education to like use this app. And I am looking for collaborators to test the app in their fields to see if we can validate it in a real life situation. And here on the right, you see our website, a website where you can also find information, and this app will be placed when it's finalized. Now, and also related to indicate that this year we produced mummyberry updates during this research, where I collected the data for the host and the Apothecia while I was doing these experiments, and produced these weekly updates to keep everybody updated on what the situation was in both Skagit and Whatcom County. And I think these were very well received, and I'm hoping to do that again this year. So with that, I'd like to thank the Washington Blueberry Commission and the Washington State Commission for Pesticide Registration, as well as the Northwest Agriculture Research Foundation for funding this work. I'd like to thank the Washington Blueberry growers for collaborating on this project, the small fruit pathology team at WSU, Jabe Shai of Oregon State University, and Michael Grant of the University of Washington, as well as the WSU Mount Vernon Northwestern Research and Extension Center for supporting me during this project. And with that, thank you for attending the session today, and please ask me questions if you have any.
Hi. Out of curiosity, and I've heard this before, but I haven't ever heard what the temp is when you're calling the cooling of the plant or the in dormancy. What is the temperature of that? Is it just, you know, in the 40s, 30s, 20s?
So the question is, what's the temperature of this chilling requirement? And this is temperatures between 0 and 7 Celsius. And I think I'll need to have some help to calculate Fahrenheit. Okay, 32 to 44 Fahrenheit. Thanks. Any other questions?
So I really liked the video you showed, and it looked like the conidia came out, or the ascospores came out all of a sudden. Is
Yes.
that real time?
Yes.
So they
Yeah,
really,
it is.
and when an apothecium releases its spores, does it release all of its spores at the same time?
It does not release the spores all at the same time. I don't think so. But when I do that myself, like I have the mummies and the apothecia in a box, it's often right when a change in the environment occurs. So when I take the lid of the box, then I will see that they go, and they will go, yeah, a couple of times, or like, yeah, they just seem to be influenced by really minor changes in its environment. Yeah.
Thanks.
Okay, thank you for Dr. Dalphy. Please help me to do thanks to Dr. Dalphy. And then for the next speaker is Dr. Alan Schreiber. He's going to talk about new product and resistant management strategies for mummyberry and botrytis in conventional blueberry. Please welcome Dr. Alan.